Hi everyone. I'm Dr. Mungli. So in today's brief lecture, I'm going to explain on glycogen synthesis and degradation in the form of animation. So glycogen synthesis, generally it will be going on whenever the person is in well-fed condition. So glycogen is basically a storage form of glucose, and it's a highly branched polysaccharide. So a glucose molecule, when it is plenty in our bloodstream, generally when we are in well-fed condition, so at that time, so when the blood glucose concentration is more than 5 millimoles per liter, so it enters into beta cells of pancreas, and thereby the signaling mechanism that will be going on within the beta cells of pancreas under the influence of glucose. So beta cells of pancreas will release insulin, and that insulin, it will help in the formation of glycogen in the hepatocyte. So the glycogen is basically, it is the linkage of glucose molecules by two types of linkages. One is alpha 1,4 glycosidic bond, which is going to link one glucose molecule with the another neighboring glucose molecule with the glycosidic bond here. As you can see here, so this particular glucose molecule is going to link with the neighboring glucose molecule with alpha 1,4 linkage. Similarly, in the straight chain here, so glucose molecule is linking with neighboring glucose molecule with alpha 1,4 linkage. Like this, the linkages will go on. And the other type of glycosidic bond that you are going to see in glycogen is the alpha 1,6 glycosidic bond. This alpha 1,6 glycosidic bond you are going to find in glycogen wherever there is branch points. Here is the branch point in a glycogen. So consider this is a linear structure, and there is a branch which is inserted here, and the glucose molecule is attached to another glucose molecule, creating a branch point by alpha 1,6 glycosidic bond. Why it is called as alpha 1,6 glycosidic bond? It's because the sixth carbon of this glucose molecule is going to link with the first carbon of the incoming glucose molecule, and that's why it is alpha 1,6 glycosidic bond. So in our glycogen, so usually there will be one glycosidic alpha 1,6 glycosidic bond with basically giving rise to a branch point for every 8 to 10 glucose residue. So 8 to 10 glucose residue in a glycogen, so there will be one branch point. So as you can see here in the left hand side here in this picture, so glucose residues which are attached by alpha 1,4 glycosidic bond, so there is a branch point here, and there is again a branch point here. Of course, in this picture you won't be able to make it out that every 8 to 10 glucose residue there will be a branch point. That is not shown here in the picture. So that is because of the space restriction that is there. So but in our cell, usually you are going to find for every 8 to 10 glucose residue there will be one branch point. So all the green colored circles that you are seeing here, these are the branch points with alpha 1,6 glucosidic or the glycosidic linkage. And also other point that you need to remember here is, so all the ends of the branches here, what are all the glucose residues that are present at the ends of all these branches, so they are all referred as basically non-reducing ends. So glycogen has got only one reducing end, and that is going to interact with the glycogen protein molecule. The line that you are seeing here, so this is the glycogen, which is a protein molecule. Now as you can see the overall structure of the glycogen here, it is highly branched. So what is the use of branching, having this highly branched glycogen structure? So there are two things to that. One is having highly branched glycogen, so it is easy for glycogen phosphorylase to degrade this glycogen within no time, because hundreds of glycogen phosphorylases, they will come over here on the branch points and start releasing one glucose molecule at a time. Basically glucose molecule will be released as glucose 1-phosphate by glycogen phosphorylase, which I am going to say later. Okay, so that's the one thing. And also while synthesizing glycogen, say person when he is there in fasting condition, so he is going to degrade this glycogen, When the person gets into fed conditions, when he gets sufficient glucose, so same this degraded glycogen can be synthesized back into normal glycogen within no time. So by having a highly branched glycogen structure, so it helps in the rapid degradation and also synthesis of glycogen. Now the glycogen synthesis, it all begins with glucose 6-phosphate converting into glucose 1-phosphate. So note that when the glucose is plenty, so initially it gets into glycolysis, and also at some point of time glycolysis is basically negatively regulated by high levels of ATPs, because sufficient ATPs indicates it's a well-fed condition. So when ATP is basically acting on PFK1 negatively, so glucose 6-phosphate accumulates, and that glucose 6-phosphate is converted to glucose 1-phosphate, and within the liver glucose 6-phosphate is converted to glucose is converted to glucose 6-phosphate by glucokinase enzyme, whereas in other peripheral tissues, so glucose is converted to glucose 6-phosphate by hexokinase enzyme. And also note that apart from liver and skeletal muscle, all the other tissues will also be able to synthesize glycogen, but at lesser quantities. So once glucose 6-phosphate is converted to glucose 1-phosphate by phosphoglucomutase, so glucose 1-phosphate is converted to UDP glucose, and this job is done by UDP glucose phosphorylase enzyme. In this process, UDP gets into the reaction and released as inorganic pyrophosphate, and UDP is attached with glucose, and that is the UDP glucose here. Now inorganic pyrophosphate is broken down into 2 inorganic phosphate molecules by pyrophosphatase enzyme, and this is an exergonic reaction, and that releases energy, and that energy is used to combine UDP glucose with glycogenin molecule. Note that glycogenin is a protein molecule, so it has got a tyrosine residue. So our tyrosine residue is tyrosine amino acid is an OH containing amino acid. It has got a hydroxyl group. So we have 3 hydroxyl containing amino acids. One is tyrosine, other 2 are serine and threonine. Okay, so over tyrosine, so UDP glucose is basically giving its glucose molecule, and UDP is released. So the glucose that is attached to tyrosine initially is coming in as UDP glucose, and glucose is attached to UDP is released, and the attachment of glucose to this tyrosine of over glycogenin doesn't need any enzyme. It is an autoglycosylation process. It has been noted that the glycogenin itself is acting as an enzyme here. So like this, few molecules of glucose will be added simultaneously, so by autoglycosylation process. Usually it will be 6 to 8 in number. So once glycogenin gets 6 to 8 glucose residues, of course it is not shown here in this particular figure, they are shown only 2, but usually it will be 6 to 8, and that is the glycogen primer synthesis. So once glycogen primer is synthesized, so now the glycogen synthase enzyme under the influence of insulin. So this is where the insulin comes into picture. So in well-fed condition, insulin will keep this glycogen synthase active. So when the glycogen synthase is active, so it takes more and more UDP glucose and attaches glucose, and UDP is released. Like this, a long linear chain of glucose residues will be formed with alpha-1,4 glycosidic bond. So like this, so when the sufficient elongation is done by glycogen synthase, so then the branching process will go on, that we'll see. So let's see the animation. So here is the glycogenin with OH residue, hydroxyl containing tyrosine residue over glycogenin. So here is your UDP glucose. Note that this is the first carbon, and here is the 4th carbon for UDP glucose. So this is how UDP glucose looks like. Okay, so there is a glucose molecule, and this is the UDP, uridine diphosphate. So autoglycosylation first. So UDP glucose is added, UDP released. So glucose is added to glycogenin. So note that this is the reducing end of glucose because it's a carbonyl carbon. So that is now participating in the bond formation attached to glycogenin. Now one more UDP glucose will come and same autoglycosylation process, UDP released and glucose is attached. So once the primer is synthesized, basically now this is the primer with 6 glucose residues attached. Of course I have shown it as 2 here because of the space. So now the glycogen synthase will bring in UDP glucose and attach it to glycogen primer. So basically elongation process is going on. So glucose is coming in and UDP glucose comes in, glucose added and UDP is released. So this is how the elongation process will go on, and all these glucose residues are now attached with alpha 1,4 glycosidic bond. Once sufficient elongation is done, usually it will be around 11 to 15 glucose residues which are added. A part of this it will be taken by branching enzyme. See, this part of the long linear chain is taken by this branching enzyme. So branching enzyme is also referred as alpha 1,4 to alpha 1,6 transferase, which is also called as 4,6 transferase. So what it does, basically it cuts off that branch in total. So in this animation I have shown individually, but generally its entire thing is cut and attached here. So alpha, basically this is carbon 1, carbon 4 and the carbon 6 of this glucose residue here. So it is now this particular area is cut and then it will be attached to the glucose residue 6th carbon here. So this is alpha 1,6 glycosidic bond. So alpha 1,4 linkages in the straight chain here, whereas at the branch point you have alpha 1,6 linkage. So wherever you see branch point there will be alpha 1,6 linkage, and furthermore all the other basically other linkages here, it is alpha 1,4 linkage. This is how the branching process will go on. So glycogen synthase and branching enzyme, they act simultaneously to produce highly branched glycogen structure. So this is how the branching process will go on. See, this is the one branch that is inserted. So then later on it is elongated by glycogen synthase enzyme. So there is this is the alpha 1,6 linkage here, and here is the alpha 1,4 linkage. So this is how the branch. So there is one more branch attached here, and that's again by alpha 1,6 linkage at the branch point. Later it is all alpha 1,4 linkage. Now we'll move on to see what happens when the glycogen degradation goes on. So the how glycogen degradation starts. So note that the glycogen that is stored in our skeletal muscle and also which is stored in our liver, so they have got different uses. So glycogen that is stored in the liver, it is meant to maintain the blood glucose level under fasting condition, whereas the glycogen that is stored in the skeletal muscle and other tissues, it is meant to give energy source, or it is one of the energy source for that particular muscle or the tissue during fasting condition. So peripheral tissues other than the liver, so they are not able to maintain the blood glucose level. So liver glycogen is the one which will release glucose from glycogen, whereas the glycogen present in other peripheral tissues, they don't release glucose, but they will degrade glycogen, and then that glucose 6-phosphate which is coming in from there, so it will be getting into glycolysis and provide ATP. So take this as an example of branched glycogen here, so which we have seen in the previous figure. So when the person gets into fasting condition, say 2 to 3 hours after meals, so usually the blood glucose level comes down and that will release glucagon. So under the influence of glucagon, glycogen phosphorylase will be activated. So at the same time glycogen synthase will be inactivated by glucagon. So glucagon at one point it activates glycogen phosphorylase, on the other side it will inactivate glycogen synthase. So what is the role of glycogen phosphorylase? So basically glycogen phosphorylase, it acts on the non-reducing ends of the glycogen with all the branches which have got non-reducing end. So it starts somewhere from here and also here. So there are several glycogen phosphorylases which will act on each branch point, and they will release glucose residue as glucose 1-phosphate. Note that glycogen phosphorylase will release glucose present in glycogen as glucose 1-phosphate. Basically, glycogen phosphorylase, it incorporates inorganic phosphate, and inorganic phosphate is acting as a nucleophile, and that's going to break down alpha 1,4 linkage and release that. So the same inorganic phosphate will be attached to glucose at first carbon, and it is released as glucose 1-phosphate. So this is how the glucose is taken out. Say one glucose taken out, released as glucose 1-phosphate, one more taken out. So the glucose residues which are present in the red circle here, or the red thing that I have marked out, so that's where the removal is going on until 4 glucose residues left at the branch point. Here is the branch point with alpha 1,4 linkage. So 1, 2, 3, 4. 4 glucose residues are left, and at that point glycogen phosphorylase cannot move ahead because there is a steric hindrance. If you can see here, so there is an L-type shape here, and this doesn't allow glycogen phosphorylase to move further because of the steric hindrance. So now it starts with the another branch here. So it is removing glucose residue, releasing it as glucose 1-phosphate, and stops when 4 glucose residues are left at the branch point. 1, 2, 3, 4. So 4 glucose residue left at the branch point, so it will stop there. So this is what glycogen phosphorylase does. So all the branched glycogen, it is acted on, all the branches are acted on by glycogen phosphorylase. Glucose is released as glucose 1-phosphate and stops at when 4 glucose residues are left at the branch point. Now what will happen to that? So over the branch point, that 4 glucose residue which are left, now the debranching enzyme will get into picture. So debranching enzyme is basically, it has got 2 enzymatic subunit. It has got 4,4-transferase subunit, and it has got alpha 1,6-glucosidase subunit. So it has got 2 catalytic mechanism. So 4,4-transferase, other name of 4,4-transferase is alpha 1,4 to alpha 1,4 transferase. So what it does is going to take these three glucose residues which are linked by alpha 1,4 linkage and shift it to somewhere else with the same alpha 1,4 linkage. It's not going to create any branch. So basically the red circle here that I have created, so that particular 3 glucose residues are taken out and attached here. Basically it is attached here. This is how the process will go on. Now this particular chain is elongated by 3 more glucose, and now your glycogen phosphorylase can come and remove these 3 glucose residue from here and stop at 4 glucose residue which are left at the branch point. Okay, so now the glucose residue that is left at the branch point, this particular glucose residue which is left at the branch point is removed by another catalytic part of this debranching enzyme called alpha 1,6-glucosidase. So alpha 1,6-glucosidase is going to remove this glucose residue and release it as glucose. Note that glycogen phosphorylase is going to release glucose in glycogen as glucose 1-phosphate, whereas alpha 1,6-glucosidase, it's going to release this 1 glucose residue left at the branch point as glucose. So it is not releasing it as glucose 1-phosphate, it is the direct glucose that is released. So it is released now. Now the branching glycogen phosphorylase will come in and remove the branches until 4 glucose residues are left at the branch point. Now the 4 glucose residue at the present at the branch point. So now this particular 3 glucose residues are taken out by debranching enzyme, that is 4,4-transferase, and reattached here. Basically it is reattached here. Now the glycogen phosphorylase will come and remove 3 glucose residue from here, and 1,6-glucosidase will come and remove 1 glucose residue left here at the branch point. So 1,6-glucosidase is removed that. Now the glycogen phosphorylase will come and remove individual glucose residue and release it as glucose 1-phosphate. So this is the complete degradation of glycogen, and now you are left with only glycogenin. So this is what happens. So approximately 1 glucose residues will be released for every 7 to 9 glucose 1-phosphate released. So you can take it as 1:9 residues are released for every branch point after glycogen degradation. So note that the glycogen degradation, it starts 2 to 3 hours after fasting, and glycogen degradation will be at peak at 12 hours of fasting, and then it will come back to baseline, or maybe it is completely depleted like this by 24 hours under standard conditions. And also note that it varies from individual to individual, but glycogen is last for 24 hours in standardized person. So that is 70 kg man with all the all the glycogen stores which are 100% full. So he can, the blood glucose will come into it will be coming from glycogen until 24 hours. By that time, after 24 hours, it is completely depleted. So person will have to depend on something else for energy needs or for the glucose needs of the body. So what happens to that glucose 1-phosphate that is released by glycogen? Phosphorylase. It all depends on in which tissue this glycogen degradation is going on. So we'll take muscle as a peripheral tissue, so it applies to even other tissues, also other peripheral tissues apart from the liver. So in this skeletal muscle and other tissues other than the liver, so glycogen that is broken down and releasing it as glucose 1-phosphate, and that is converted to glucose 6-phosphate. Now this glucose 6-phosphate, it is getting into glycolysis, and if the oxygen is sufficient, if the cell has got mitochondria and all the sufficient oxygen, so TCA cycle will go on and releases ATP and carbon dioxide. Ultimately glucose 6-phosphate is releasing 6 carbon dioxide there and releasing energy. Whereas if sufficient oxygen is not present, so glucose 6-phosphate gets into anaerobic glycolysis and releases lactate. Whereas in the liver, so the glycogen that is releasing glucose as glucose 1-phosphate, and that is converted to glucose 6-phosphate, and the glucose 6-phosphate is converted to glucose by a special enzyme which is present in the hepatocytes and also partly in the kidney. So this is a speciality of the liver because it has got this glucose 6-phosphatase which is not present in any other peripheral tissue except for the kidney. So glucose 6-phosphatase converts glucose 6-phosphate into glucose, and that glucose gets into bloodstream, and that glucose is carried to the other peripheral tissues for energy needs. So this is how the energy needs of the body will go on. So glycogen present in the liver is the one which will contribute to blood glucose under fasting condition, and that this is how glucagon is trying to maintain the normal blood glucose level under fasting condition if it occurs within 24 hours. So that's the one which is coming from glycogen. After 24 hours or maybe after 12 hours of fasting, so there are other sources of glucose for our body, particularly for erythrocytes and the brain. So that's the fate of glycogen. So that's how glycogen is synthesized and glycogen is degraded. So if you have any questions, you are most welcome to drop your questions in comments below or send an email to my email ID. So thanks for watching. See you again in my next presentation.